Okay, this is the product, you know it. Now what happens then? Pyruvic acid enter mitochondria. In the mitochondria, is the first step where carbon dioxide is produced. Where carbon dioxide is produced. And the enzyme is pyruvate dehydrogenase complex. Now question comes, why the term complex is used here? Most important thing is why the term complex is used here. Remember this thing, this reaction takes place through a series of activities. First of all, pyruvic acid plus TPP, thiamine pyrophosphate, forming hydroxyethyl TPP with the release of carbon dioxide. The first step. Next step, hydroxyethyl TPP lipoamide oxidized to form acetyl lipoamide and TPP becomes free. After that, acetyl lipoamide then reacts with COSH, forming acetyl coenzyme it is reduced lipoamide oxidized and quickly becomes after that so for the products here, look, the products are so these are the products you get over here. Clear? This is how actually this reaction takes place. In the first reaction, when pyruvic acid is converted to hydroxyethyl TPP, here the pyruvate dehydrogenase acts. It is the decarboxylase activity of dehydrogenase is found here. Decarboxylase activity of dehydrogenase is found here when pyruvic acid is converted to PDH. PDH means pyruvate dehydrogenase. When acetyl lipoamide is formed, here we get the decarboxylase activity of dehydrogenase. Here here decarboxylase activity of dehydrogenase is found. Here decarboxylase activity of dehydrogenase enzyme, PDH enzyme or PDH enzyme is found. Whenever hydroxyethyl lipoamide is converted to acetyl dihydrolipoyl transacetylase. Space is not there. To form transacetyl lipoamide. Actually, these two reactions cannot be this. Both of these are the same enzyme, dihydrolipoyl transacetylase. After that, lipoamide will reduce to form ritomide. Is here we will get dihydrolipoyl dehydrogenase. So this is what about the different enzymes. You need not to panic about this. Here it is said why it is called pyruvate dehydrogenase complex. First reaction, pyruvic acid is converted to hydroxyethyl TBP, and here the decarboxylase activity of PDA is found, because the pyruvate dehydrogenase is found. The next reaction and second two reaction. In one, hydroxyethyl TBP is converted to acetyl lactamate, and then it is converted to acetyl coenzyme. Both the reactions are catalyzed by dihydroalcohol transacetylase, and acetyl group is transferred. And when reduced lactamate is converted to oxidized, here hydrogen is removed out to FAD, so it is dihydroalcohol dehydrogenase. So these are the enzymes taking part here. Clear? That's why the term used pyruvate dehydrogenase complex, because the reaction is actually taking place in this way. We are in short, we are showing this. Clear?
Yes, sir.
Yes, sir.
So last step কোনো enzyme নেই?
No, not required. The same way. A dihydroalcohol dehydrogenase catalyzing both actually. Same enzyme is acting here also. It is taking place simultaneously.
স্যার COASH এর ফুল ফর্মটা কি?
Acetic COAS is together called acetyl coenzyme. Wait a minute. COAS is together called coenzyme A. Coenzyme A. Clear? If you write COA, then also it means COS actually. Are you getting me? Can I rub it out now?
Yes, sir.
Okay, sir.
After that, acetyl coenzyme, you can call it this is the intermediate step between glycolysis and Krebs cycle. After that, what happens next? Observe acetic acid. Forming so this is the first reaction of Krebs cycle. Six carbon compound. Citrate synthase is the enzyme. And if you go to the citric acid, what it is found actually, CH3, COS, this is actually the acetyl coenzyme used to react with oxaloacetic. Look this, and eventually forming the citric acid with 3 carboxylic group. Look, this is the citric acid.
স্যার কার্বন
There are
সালফার
how many
বন্ডটা কি
carboxylic group are there? 1, 2, 3. That's why tricarboxylic acid or TCA. So the first stable product of the oxaloacetic acid OAA is acetyl coenzyme. Look at this. The first reaction, a 2-carbon acetyl coenzyme reacts with actually 4-carbon compound oxaloacetic acid, and it is a kind of condensation reaction, and the enzyme is citrate synthase forming a 6-carbon compound as citric acid, the first stable product having 3 carboxylic group. As citric acid possesses 3 carboxylic group, it is also called tricarboxylic acid or TCA. Clear?
স্যার এসিটাইল কোয়ের মধ্যে যে কার্বন সালফার বন্ডটা এটা কি এনার্জাইজড বন্ড?
No, yes. Oh,
স্যার স্ল্যাশ দিয়ে রাখা যায়।
oh, that's less not having, not. Clear?
ইয়েস স্যার।
Okay. Is it clear now? Then comes the next reaction. I'm rubbing it out, right?
Yes, sir.
Now in the next reaction, what happens? Citric acid is a stable compound. Here, from here, hydrogen is required to be to facilitate oxidation. Just a minute. In Krebs cycle or citric cycle also, oxidation takes place by means of dehydrogenation only. Again, you can say that in no step of Krebs cycle, oxygen takes part. So here also, it occurs by dehydrogenation. That's why the citric acid is a stable compound. So that from it, the hydrogen cannot be removed out. So some change in the structure is required in the structure of citric acid. How? You see. At first, from citric acid, a molecule of water is released out from C-saponitic acid. Water. Aconitis. Next step. C-saponitic acid, same enzyme. So this is the reaction. So what happens in this reaction? Look. From OH is coming out here, then ultimately CH-COOH. So this is what formed first. After that, when water is added, what the change occurs? Change occurs here. So OH and H added here. Now, this carbon is most unstable now, can release hydrogen. This is called isocitric acid. So why? Because now, the next reaction you will see now, this is the carbon which is more unstable, from where hydrogen can be removed out to facilitate oxidation. That's why such conformation occurs. The same thing happens, only water is added. Water is removed, then water is added. The same thing is, but there is some change in the arrangement, so that now it can take part in the oxidation reaction. Is it clear to you? This is the cause of these two reactions. The
Sir, ये last part repeat करो.
And yes.
Sir, last part एकटू repeat करले भालो है. Isocitric acid part.
You see, in isocitric acid what happens? You see, hydrogen is added here. Hydrogen is OH is added here. Now, whenever a class carbon OH is there, now you see these two. Now it's almost unstable, so that they can donate this hydrogen. From this form, you cannot donate hydrogen to the NAD. But here, this form is suitable for donating hydrogen to NAD, so that it can take part in the oxidation. Actually,
Okay,
so that citric acid can take part in the oxidation reaction, these changes are taking place. Are you getting me now?
Yes, sir.
Now I have to rub it out again. Clear? Clear?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. After that, in the next reaction, you see isocitric acid plus NAD plus first reaction is irreversible. Then forming NADH by H plus forming oxalosuccinic acid. Enzyme is the substrate isocitrate. As hydrogen is removed out, dehydrogenase. But oxalosuccinase is very unstable, I am telling you. But I will be showing you after this reaction. Is it clear now? What happens here? How does it occur? CH2COOH COOH. So from here, these two hydrogens are removed out, these two. So that you will get CH2COOH this is the oxalosuccinic acid. This is what called oxalosuccinic acid. Clear? But this product is, the enzyme is isolated here, but the product is temporary. Before leaving the active side of the enzyme, oxalosuccinic is from oxalosuccinic acid, a molecule of carbon dioxide is released out, forming alpha ketoglutarate acid. I am repeating it again. Suppose you see, this is what the suppose isolated dehydrogenase. Now, this is the active site. So this is for the enzyme, the substrate isocitric acid. This is the isocitric acid binds to the active site. What happens next? Isocitric acid is converted to oxalosuccinic acid. And before leaving the active site, from this enzyme, a molecule of carbon dioxide is released out, and the product would become now alpha-ketoglutarate acid. So all these are same isocitrate dehydrogenase. It is also isocitrate dehydrogenase. Now I am showing you this. I can rub. Is it clear to you what happens here? It is clear after the formation of oxalosuccinic acid.
Yes, sir.
Yes, sir.
Okay, now I am rubbing it out. Look what happened here. Oxalosuccinic acid alpha-ketoglutarate acid with the release of carbon dioxide. First thing, then why same enzyme it is shown here? Is it whether this diagram is clear? First of all, isocitrate dehydrogenase is the active site where isocitric acid binds. The enzyme product complex is formed forming oxalosuccinic acid. Before leaving the active site, from each one molecule of carbon dioxide is released out, forming alpha-ketoglutarate acid. This is the what is this product called? Alpha-ketoglutarate acid. That's why same enzyme is found to catalyze both these reactions. So that's why same enzyme is found to catalyze both these reactions. Clear?
Yes, sir.
Okay.
স্যার
Yes.
স্যার এখানে তো কার্বন ডাই অক্সাইড রিলিজ হওয়ার পরেই অ্যালফা কিটোগ্লুটারিক অ্যাসিড প্রডিউস হচ্ছে।
হ্যাঁ।
আচ্ছা আচ্ছা ঠিক আছে।
বুঝছিস তো?
হ্যাঁ।
Normally what happens? Substrate binds to enzyme substrate complex is formed. Then by the activity of enzyme it becomes a product, so enzyme product complex is formed. Now product is found to be released out from the active site. Okay. But here what
Yes, sir.
happens, before leaving the active site, suddenly one molecule of carbon dioxide is released out. So it is no more, with the release of one molecule of carbon dioxide, it is no more Hausdorff succeeding acid now. Clear?
Yes, sir.
This is what, that's why the same enzyme. So you can go through it here like this. CH2COOH, CHCOOH, so this is the product with the release of carbon dioxide. It is this carboxylic group is released as carbon dioxide, so you get here like this. So it becomes CH2 now. Clear? This is what happened here. After that, COSH. So that again oxidation occurs, forming succinyl coenzyme. Plus again there is release of carbon dioxide, and you know it's a 4-carbon compound. So it is only 5-carbon compound formed in Krebs cycle. Remember this thing. Hausdorff success alpha-ketoglutarate is the only 5-carbon compound formed in Krebs cycle, and first 4-carbon compound found is succinyl coenzyme. If you go to the structure, what happens you see, here from here one carbon dioxide is released out again. So you will get COS, COA again. This is succinyl coenzyme. So what happens here? From here, one carboxylic group is released out, and it is reduced by this hydrogen, and also the hydrogen present in the COH. So this is we get here. Enzyme is alpha-ketoglutarate dehydrogenase. Okay, one thing. These are both are irreversible. After that, succinyl coenzyme converted to succinic acid, and in this reaction, what happens? GTP to form GTP, and this GTP then this is what happens here. Or you can write it here like this. Enzyme is thiokinase so here substrate level phosphorylation occurs here. This is the step where ATP is produced actually. Clear?
স্যার রিঅ্যাকশনে স্যার জিটিপি প্লাস এডিপি স্যার কিভাবে হয়েছে?
From GTP, phosphate group is transferred to ADP. By getting the phosphate group from GTP, the ADP becomes ATP. Are you getting me?
ইয়েস স্যার।
Enzyme is here, enzyme is required. Enzyme involved here is wait. This is the reaction. Enzyme is nucleoside diphosphate kinase. Clear after this? And the structure I am showing here. You see, succinyl coenzyme CH2 then in case of succinic acid the same thing happens. So CH2COOH and CH2COOH is formed actually. CH2COOH this is the succinic acid. That means CH2 the CO is formed here. So here for condensation, 1 molecule of water is actually required. Tricarboxylic acid, hence it is also called TCA cycle. Remember this thing. Now, secondly, most important thing here that do you know in no step of Krebs cycle, you see in no step of Krebs cycle, oxygen takes part, but Krebs cycle cannot be operated in absence of oxygen. Are you getting me? If you go to all steps of Krebs cycle, in no step of Krebs cycle, oxygen takes part, but in absence of oxygen, Krebs cycle cannot be operated. Can you tell me why? But in absence of oxygen, Krebs cycle cannot be operated. Can you tell me why?
Sir, huge amount of energy is required for Krebs cycle.
No energy now. Basically, one thing I'm telling you is if you go to all steps, in no step oxygen takes part. In no step oxygen takes part, but Krebs cycle cannot be operated in absence of oxygen. Question is why?
স্যার এখানে NAD and FAD শেষ হয়ে যাবে NAD and FAD শেষ হয়ে যাবে মানে অক্সিজেন না থাকলে এখানে NAD and FAD চার্জ উঠে যাবে
NAD কি হয়ে যাবে একটু পরিষ্কার করে বলো
বলছিলাম অক্সিজেন যদি না থাকে তাহলে NAD এবং FAD শেষ হয়ে যাবে
কেন
পেয়েছি এখানে
হ্যাঁ
এখানে পেয়েছিলাম আর কি
তো বুঝতে চেষ্টা করোনি কেন look one thing I am telling you in Krebs cycle oxidation occurs by means of dehydrogenation and to facilitate dehydrogenation the coenzymes like NAD and FAD are required to be present in their oxidized form. I am repeating it again. In Krebs cycle, oxidation of substrate occurs by means of dehydrogenation. And to facilitate dehydrogenation, different coenzymes like NAD, FAD are required to be present in their oxidized form. Okay. Oxidized form means, NAD plus this is the oxidized form and this is the reduced form. If Krebs cycle is to be carried out, they are to be found in oxidized form. Look here, oxidized form. In Krebs cycle, NAD, FAD, etc. get reduced to form NADH plus and FADH plus, which are the reduced form of coenzymes. And these reduced coenzymes can be converted to their oxidized form only during electron transport in the presence of oxygen. So if oxygen is not available, NADH and FADH cannot be converted to NAD and FAD. So that Krebs cycle cannot be operated. Is it clear now? দেখো যদি বুঝতে পেরেছো কেন?
হ্যাঁ স্যার।
Because you know, oxidation of reduced coenzyme takes place in the presence of oxygen only. Remember this thing. In the electron transport chain, in the presence of oxygen, the oxidation of this reduced coenzyme takes place. So if oxygen is available, then no problem. In the electron transport chain, in the presence of oxygen, reduced coenzymes are oxidized. Clear? But if oxygen is not available, reduced coenzymes produced cannot be converted to their oxidized form. If coenzymes are not available in their oxidized form, Krebs cycle cannot be operated. And hence it is said, although in no reaction of Krebs cycle, oxygen takes part, but Krebs cycle operates only, but Krebs cycle operates only in aerobic condition. That is when oxygen is available. Clear to you all? Or any doubt?
Okay, sir.
ওকে স্যার।
So you need to write the Krebs cycle operates only under aerobic condition. Then already I discussed the unidirectional reactions of the soap. Another most important regulation. Can I rub it out? It's okay. I'm not rubbing it out. Regulation of Krebs cycle. Three enzymes following three enzymes regulate Krebs cycle. The enzymes are citrate synthase, then comes to isocitrate dehydrogenase, and lastly the alpha-ketoglutarate acid dehydrogenase. So, how they can regulate? Citrate synthase catalyzes the formation of citric acid from acetyl coenzyme and catalyzes the formation of citric acid converts acetyl coenzyme, reacts with acetyl coenzyme and oxaloacetic acid. Then actually isocitric acid is converted to alpha-ketoglutarate acid in the presence of isocitrate dehydrogenase. And then alpha-ketoglutarate dehydrogenase catalyzes the formation of succinyl coenzyme from alpha-ketoglutarate acid. Now remember this. Besides these three enzymes, availability of adenosine diphosphate also play an important role in regulating the Krebs cycle. Besides these three enzymes, Besides these three enzymes, availability of ATP. Availability of ATP is very important for দেখো secretary cell to proceed.
স্যার নিচে জায়গাটা দেখা যাচ্ছে না।
হ্যাঁ দেখা যায় না ও আচ্ছা তাই আমি অন্য জায়গায় লিখি beside these three enzymes দেখা যাবে beside these three enzymes of ADP is very important to proceed for citric acid cycle to proceed. Is it visible now?
Yes, sir.
This is what about the Krebs cycle. And you need to remember the whole cycle and also the regulation of this, why it doesn't occur in the presence of in absence of oxygen, and why Krebs cycle is called citric acid cycle. And besides you, so there are some products of this cycle may be utilized in the formation of other substance. So the alpha-ketoglutarate acid may be utilized in the synthesis of glutamic acid. Alpha-ketoglutarate acid may be utilized in the synthesis of glutamic acid. So there are some intermediates formed in Krebs cycle may be utilized in the synthesis of some other compounds. Remember these things. Okay, like as I said earlier, alpha-ketoglutarate acid is utilized in the synthesis of what is called glutamic acid, and then oxaloacetic acid. Sexual coenzyme is used for the synthesis of heme and porphyrin of hemoglobin. Oxaloacetic acid produced may be used in the synthesis of aspartic acid. So there are some intermediates which are utilized in the synthesis of some other compounds. Like as I said, alpha-ketoglutarate acid to make glutamic acid, oxaloacetic acid to make aspartic acid, succinyl coenzyme to make porphyrin and heme of hemoglobin. And as you know, the Krebs cycle is also called amphibolic pathway because here there is both anabolism and catabolism is found. Essentially because it gets catabolized simultaneously, you also need to remember that here some intermediates are also taking part in anabolic phase. As both anabolism and catabolism is found, so the term used is amphibolic pathway. Clear? Is it clear why it is called amphibolic?
Yes, sir.
বুঝতে পেরেছো কেন amphibolic বলা হয়? Because some compounds produced in this cycle, they are actually taking part in some anabolic pathway. Example কি? Suppose alpha-ketoglutarate acid is involved in the synthesis of glutamic acid. So it is an anabolic pathway. Oxaloacetic acid is involved in the synthesis of aspartic acid. It is also an anabolic pathway. So there is a mixture of both anabolism and catabolism here in the Krebs cycle. That's why it is also called amphibolic pathway. Clear?
Yes, sir.
Excellent. Yes? Oh, next topic. This is the next topic. Is it clear up to this?
Yes, sir.
And you need to remember the reactions, in which reactions CO2 is produced, in which reactions reduced coenzymes are produced. Okay?
Okay, sir.
Okay. In my next class, I have to discuss about the actually terminal aspiration.
স্যার
Okay.
একটু সাইড দেবে
আচ্ছা দাও একটা মিনিট পরে না আমি আসছি জাস্ট মিনিট Okay, completed.
Yes, sir.
Yes, sir.
বলো
স্যার অ্যানাবলিক আর ক্যাটাবলিক রিঅ্যাকশনের একটু এক্সাম্পল বলেছিল, এটা একটু বলতে পারবে?
ওই যে alpha ketoglutarate acid, the intermediate form in Krebs cycle may be utilized in the synthesis of glutamic acid. Clear?
Yes, sir.
Then you need succinyl coenzyme and another intermediate may be utilized the synthesis of porphyrin and heme. Oxaloacetic acid may be utilized in the synthesis of aspartic acid. Clear?
Yes, sir.
Okay, thank you. That's all for today. Okay, I'm going to conclude now.